biologists. Today we're going to talk about pedigrees and we will do some practice questions. Pedigrees are used to track a trait going through a family. And so we're going to denote different generations and male and female and whether or not someone has the traits. If you are a female, you'll be shown as a circle. And if you are a male, you'll be shown as a square. If you are not filled in, you do not have that trait. And if you are filled in, you do have the trait. If it's crossed through the circle or the square, that means that that person is deceased. Sometimes that's not shown and it shows a full pedigree that's like 7 generations long and you know they're not all still alive. But if you do see that, it means that there is a deceased person in the family. When a male and a female have children, they will put a line between them, and then the line will drop down and branch off to each of their offspring. So that's how we will know that people had kids. Pedigrees are not used to track emotional relationships. It's just tracking our genetics. So we're only caring if there's been offspring. The generation number is noted in Roman numerals. So 1 and 2, so this is the P generation and the F1 generation. And the individuals in that generation are denoted with Arabic numbers. So the visual 1, 2, 3. So this couple right here had 3 children, 2 boys and 1 girl. In general, it usually goes from oldest to youngest as well, but that might not always be the case. So let's look at a pedigree. We can visually identify what type of inheritance is being shown based on the pattern of affected people in a pedigree. So our first pedigree is a dominant trait. I know that this is a dominant trait because I have an affected parent and I have affected offspring in every generation. So let's look at the genotypes being shown here. I have my father and I have my mother. My mother has whatever this trait is that is dominant. She has at least 1 capital allele. And I know that because she's expressing the dominant trait. She is shaded in. Since this is a dominant trait, I also know the father's genotype. To be expressing the recessive phenotype, he must have 2 recessive alleles. So here's his genotype, little a. This couple had babies, and they had 1, 2, 3 children. So whatever number of kids are attached to the parents using these lines, that's how many children they had. So they had 2 girls and a boy. Of their offspring, they had a girl who does not express the dominant trait. So that means that this individual must be homozygous recessive. They have a son that has the trait. That means that he inherited this dominant allele from mom. I know he got it from mom because dad does not have a dominant allele to give. Because this coupling is a homozygous recessive and at least 1 dominant allele. Now, I don't know if she's homozygous dominant or heterozygous by looking at her, but I do by looking at her children. Because she had a daughter with no dominant trait, that means that this mom had to have a little allele to give this daughter. So looking at the offspring allowed me to fill in the genotype of the mom. I also know that because this son got the big A from mom, he also must be heterozygous because dad only has little a's to give him. Let's look at the last daughter. She is expressing the dominant trait, so she inherited that big A from mom, and she inherited a little a from dad because that's all he has to give her. Cool. Now let's look at the daughters of the firstborn girl in generation 2. So she coupled and she had 2 daughters. Neither of those daughters have the trait. That means that both of these daughters are homozygous recessive. This son partnered with a person and they had 2 kids, a boy and a girl. Both of those kids have the trait. So they inherited that big A from their father. I know it came from their father because their mother is not expressing the trait, which means that she must be homozygous recessive, and both of their kids are going to be heterozygous. Now here, this last daughter, she had two sons, and one of those sons inherited her dominant allele, but the other did not. The father is not expressing the trait, so he must be homozygous recessive, and that means that he donated a little a to both of those sons, and this is the genotype of everyone on the pedigree, including this person that we didn't add, but because they're not shaded, we know that they are homozygous recessive. My indicator that this was a dominant allele is because it showed up in every single generation, and there was never an offspring that had a trait that the parents didn't already have. So we'll look at another example of a pedigree. And this time, we will look at a recessive trait. I know this is recessive because all of a sudden, I have an offspring who has the trait from parents who do not. So that is my indicator that this is a recessive trait. If this is a recessive trait, I know the genotype of these offspring. They're expressing the recessive phenotype. So they both must be little a, little a. Because they're both little a, that means that both of their parents have to have a little a to give them. But their parents are not expressing the recessive phenotype. So both of their parents must be heterozygous. If we go back to the P generation, we know that there's a heterozygous son. That means that one of these parents needs to also be heterozygous. But the other one, we don't know if they are homozygous dominant or if they are homozygous recessive. We don't know because we don't have enough information to fill that out. Just having one heterozygote parent would be enough to produce an offspring that's also a heterozygote. So one of them is going to be heterozygous. One of them could be homozygous dominant, but we don't know which is which. Cool. We could figure it out, though, if they had more children. If they had more children, maybe eventually they would have an offspring that's affected. And if they did, we would know that they were both heterozygous, because then we would know that they both had the recessive allele to pass on to their offspring. All right, and the last one we'll look at is for a sex-linked trait. So a sex-linked trait kind of looks like a recessive trait because we're having offspring that are affected from parents that are not. So it's going to be a recessive trait. But look, it's only men that are affected. And so that's our indicator that this is a recessive sex-linked trait. Let's look at this individual right here. So generation 2, individual 3. This individual has the shading. So this person is affected. They're going to have the recessive allele on their X chromosome. Because this is a male, they're also going to have a Y chromosome. Every square will have one Y. So we could fill in the Ys that we wanted to. And those Ys will not have the allele on it because it is a sex-linked trait. It is only on the X chromosome. That Y chromosome is too small. It's not going to hold the trait. That means that this person inherited the recessive allele on the X chromosome from one of their parents. I know that it's going to be from mom because dad already gave the Y chromosome. So he inherited his Y chromosome from the dad, not the X chromosome. That's what made him a male. So that means that mom has to have at least one little recessive allele, but because she herself is not expressing the recessive phenotype, I know that she is heterozygous for this trait. Let's say this is color blindness. So she is a carrier for the color blindness gene. but she is not expressing color blindness because she has the dominant allele, which allows her to see color. When they had an offspring, she passed on that gene to her son, causing him to be colorblind. The dad is not colorblind, so I know that he has the dominant allele as well. So those are the genotypes of my parents, and I can tell those genotypes because I know that a filled in square is going to be an affective male, and that it's a X-linked trait because only males are affected. Because only males are affected, I can go ahead and fill in the genotype of every affected male because they must have that little recessive allele to be expressing color blindness. And I know that any male that does not have color blindness, who is not shaded, must have the dominant allele because they are expressing the dominant phenotype of non-color blindness. So I can fill in all of those males' genotypes. Now, when we look at our females, we could figure out their genotypes based on their offspring. So here in generation 3, individual number 5, this couple had 2 kids. One of their kids is a colorblind male. That Y had to come from dad. Mom doesn't have a Y to give. So that means that this little b had to come from the mother. Because the mother is not expressing colorblindness, I know that this mother has one of each of the colorblind allele. And so she is heterozygous. She's a carrier, but she's unaffected. When we look at the daughter, individual 6 in generation 3, this daughter is going to get 2 X chromosomes, one from dad, one from mom. Dad is going to give this daughter his dominant allele. So she is not colorblind, but mom could be donating either or. So it could be big B or it could be little b. We don't know, and we won't know until this person reproduces. And if they had a child that was a male and did have color blindness, then we would know, that's coloring it in, we would know that this X chromosome had to exist because it got passed on to this kid. But we don't know that until if this daughter has children, and whether or not a boy is colorblind is not always the case. So if she had a boy without color blindness, it wouldn't tell us anything. We'd have to see if color blindness continued to determine her genotype. Let's look at this mom. So here in generation 2, individual 1, she is going to have a offspring, a daughter. Her daughter is going to have a son that is colorblind. I know that that little X with the little b, the colorblindness gene, had to come from mom because the Y chromosome came from dad. So this individual here has to have at least one little b, but because she is not colorblind, I know that she also has one big B. That one big B must have come from her father because that's the only X he has to give her, and he has to give her an X. So that means that one of her little b's had to come from her mother, but her mother is also not colorblind. So that tells me that her mother's genotype is heterozygous. So this little X here had to come from mom because dad only had the dominant trait to give her. And in that way, you could go along and you could fill in every genotype on the pedigree, unless you can't fill it in because you don't know whether or not they are a carrier or homozygous dominant. The SLC45A2 gene controls the difference between white and orange tigers. Orange is dominant over white. Which of these genotypes could this tiger have for the SLC45A2 gene? Go ahead and pause it and ask yourself, and then I'll give you the answer. Because this tiger is expressing the recessive phenotype, it must be recessive, homozygous recessive. This tiger's genotype is little t. That means that this tiger is homozygous recessive. Is it possible for these two tigers to produce the two cubs shown below? Create a Punnett square to support your answer. What we're looking for is whether or not we could get a homozygous recessive offspring. That's the offspring we're looking for here. From a homozygous recessive parent and at least a dominant having trait parent. And the answer is yes, but only if this orange tiger is heterozygous. If this orange tiger is homozygous dominant, then no, you would not have any of these white tiger babies. How many copies of the SLC45A2 gene would be found in this tiger's somatic cells? Tigers are mammals. Mammals are diploid. There would be 2 copies of the gene. How many copies of the gene would be found in this tiger's sperm? Sperm are haploid. There will be 1 copy. If we assume that this tiger's genotype is heterozygous, what genes could his gametes contain? Each gamete is going to get 1 of the alleles, so he could produce gametes with a T capitalized or a T lowercase. Here is a comic for you. We've got 2 fish. They have different color stripes or different pattern stripes. And then we have a little fish in front of her and she says, "Meet my mom and dad." Based on what you know about biology, do you think that the fish introducing her parents behind her could have inherited her pattern? Explain your opinion using biovocab. This would be a great example of codominance. Both of the parents' genotypes are being expressed in the little fish. All right, if you have any questions, email me and I will talk to you soon. Max and Parker say hello. Have a great rest of your day.